Great. Thank you, everyone. It's such an honor and a delight to be presenting to the constituents of Fighting Blindness Ireland. I'll be going over some of the highlights in retinal research going on right now. A lot of clinical trials, some emerging therapies that are moving into clinical trials. And I guess this is a good thing, I won't have time to cover everything. So I'm happy to follow up with any of your questions. And you can move on to the next slide. And as Laura said, I had the real pleasure of visiting Dublin last year or 2 years ago at this time. And I got to see a lot of great sites, Cliffs of Moher and the Trinity College, Newgrange. But one of the great little finds that I had was I visited Howth. Am I pronouncing that correctly, Howth?
You are, yeah, Howth.
Okay. Yeah, I accomplished something this morning. Anyway, I stumbled across this little museum called the Hurdy-Gurdy Vintage Radio Museum. It's on a hill. It wasn't on any map that I had, and it's this small circular building that's packed with all these old-time radios from World War I, World War II, and the gentleman there just for about an hour described all the different radios and the history of the museum. So it was a nice little find that maybe not all of you know about. So if you're ever in Howth, I strongly encourage you to look up the Hurdy-Gurdy Vintage Radio Museum. Okay, well, let's get into the research here. We can go to the next slide. So when I present research, I often like to give context for where the research is at, sort of from the big picture. And there are at least 40 clinical trials currently underway across the US and Europe and other parts of the world for emerging therapies. And this number continues to increase every couple months. We get a new clinical trial, and I know there's going to be another announcement of a clinical trial in another day or two. The company that I'm thinking of has not formally announced it, so sorry, I can't say what that will be. But over the past 12, 13 years, we've gone from just a handful of clinical trials to now over 40. So it's a very exciting time. And again, I'll be covering many of these. And in addition to the clinical trials, there are many, many more therapies that are in the clinical trial pipeline that are a year or two away from moving into human studies. So this momentum will continue to build in the coming years. And what's exciting for us, and I'm sure Fighting Blindness Ireland, is the research that we're funding is attracting outside investment from companies. And you'll see in many of my slides references to companies that are starting up to get these therapies into clinical trials. And then there are bigger companies, big pharmaceutical companies and biotechs that are then licensing the technologies or acquiring the companies to move them all the way through the clinical trials. You have to keep in mind, a clinical trial costs hundreds of millions of dollars to complete. It may cost a few million or 10 or 20 million to get it into the clinical trial, but to get it through the trial, you need hundreds of millions of dollars. So it's really important to attract those outside investments. So let's start off talking about gene therapy. We can move to the next slide. So just a quick overview of what gene therapy is. And I'm guessing that many of you understand that in the inherited retinal disease space, these conditions are all caused by mutations in a single gene. So for just about everybody affected, there's only one gene that's at the root cause of the problem. Now, there is a challenge in that there are more than 270 genes, each of which can cause a retinal disease. So it's a very diverse group of rare conditions. But what researchers envisioned decades ago was delivering new copies, healthy copies of the gene in question to replace the mutated gene. So replace the bad copies with good copies. And that's what's worked really well thus far in human studies. And the way that's being done in most cases is that researchers are engineering viruses. They're like containers to deliver the genes into retinal cells. And in this image on the right, this knobby, colorful ball is a viral container. And in a gene therapy, there are literally millions and millions of these containers to deliver the needed gene to the retina. And we use viruses because they're very adept at penetrating cells of the retina. Now, these viruses, as they naturally occur, are safe, and they're engineered for therapies, they're not in their native form. So these are very safe. Obviously, when you hear virus these days, we get a little nervous. But again, these are very, very safe. So that's the basic concept behind gene therapy. And I'll qualify that further by saying this is gene replacement therapy. There are other ways you can use gene therapy besides replacing the bad gene with a good gene, and we'll be talking about those in a few slides. So let's move on to the next slide. So I'm guessing that many of you know about Luxturna. It's the first gene therapy that received regulatory approval for the eye or any inherited condition. It's approved in the US by the FDA and other regulatory agencies in Europe and other parts of the world. This is for people who have mutations in the gene RPE65. This is one gene. But the therapy in clinical trials, and now that it's commercially available, has had outstanding results, especially with younger kids. These kids have been able to put away their canes. They could see stars in the sky for the first time. Many are seeing their parents' faces for the first time. So very dramatic results. This came out of labs in the US, Children's Hospital of Philadelphia and University of Pennsylvania. I'm proud to say that Foundation Fighting Blindness in the US funded research for those early studies. It moved into clinical trials in 2017 at Children's Hospital of Philadelphia. CHOP spun off a small startup company called Spark Therapeutics in 2013. The clinical trial was already off the ground then. And then about a year ago, Roche Pharmaceuticals acquired Spark Therapeutics for, I think it's somewhere in the neighborhood of $4 billion. And I think the most important takeaway in talking about Luxturna, not only is it helping that subset of people with RPE65 mutations, but it's really provided affirmation. It's opened the door to using gene therapy to treat inherited retinal diseases. It's affirmation for companies and researchers that this is a good way to go to save and restore vision. So let's talk about some of the other clinical trials that are underway. So we can go to the next slide. So one company that has a couple of clinical trials underway is Biogen, and their choroideremia gene therapy is in a phase 3. That's worked impressively in earlier phases, and they've completed enrollment in the phase 3. So when they get the final data from this phase, they'll be hopefully ready to move on to the regulatory agencies. So if you look back on what's happened with choroideremia, there was important research funded at University College of London. Then University of Oxford launched a clinical trial, spun off a company called Nightstar, and then about a year ago, Biogen acquired Nightstar. So this is a similar trajectory to Spark. And it was that early lab funding that got things going. Now, Biogen, along with two other companies that I'll be talking about, also has a gene therapy in the clinic for X-linked RP. And they've recently moved into a phase 2/3, which again is the last phase before they can potentially move into or submit the therapy application to the regulatory agencies. And there's been some visual improvements in this trial as well. So our fingers are crossed. Next slide, please. So another company in the US that's been doing well in the gene therapy space is called Applied Genetics Technology Corporation, or AGTC. And they too are moving their X-linked RP gene therapy into a phase 2/3. They've shown improvements in visual acuity and retinal sensitivity. Their trial at this point is just in the US. I honestly don't know if they're planning on launching some sites overseas in Europe for their clinical trial. We'll have to wait and see. AGTC also has clinical trials underway for a condition called achromatopsia, which, because it affects cones give us vision and daylight settings in bright conditions. Because achromatopsia affects cones, it causes day blindness. People can't see in light very well. It's kind of the opposite of RP. Those trials are at an earlier stage. They have had some encouraging results, but it's early. So we're waiting to hear about more data coming from their achromatopsia trials. Now, AGTC is also pursuing clinical trials for dry AMD and Stargardt disease. Those have yet to move into those clinical trials, so stay tuned there. The last thing I'll say is AGTC is a very close partner of the Foundation Fighting Blindness here in the US. So yet another company coming out of the UK, MiraGTx, is partnering with Janssen also on some X-linked RP and achromatopsia clinical trials. MiraGTx also had encouraging results for their X-linked RP gene therapy in phase 1/2. They are also moving into phase 3 as well. They do have sites in the US and a couple of sites in the UK as well. They as well have achromatopsia gene therapy clinical trials underway and have yet to report data there. Those are at an earlier stage, so stay tuned there. So let's move on to another slide, please. So I've given you some highlights of the trials that have moved further along. There are several other trials that are at an earlier stage or soon to be moving into the clinic. Harama, a company in France, has a clinical trial underway for RP for people with mutations in PDE6 beta. I'll note that they're working with Nijmegen in the Netherlands to launch a clinical trial for people with CRB1 mutations. Mutations in that gene also cause LCA. Novartis in Sweden has a clinical trial underway for RP caused by RLBP1 mutations. Another trial underway here in the US at University of Pennsylvania is for people with GUCY2D mutations. There's a little startup company that we're funding called Itsena that's come out of Shannon Boy's lab out of the University of Florida. And again, that trial is underway. Now, some other preclinical work that's going on, hopefully to move into the clinic soon. This is by no means everything, but just some examples. There's a company called Iveric Bio who's pursuing clinical trials for rhodopsin mutations, which cause RP, Best disease, Stargardt disease, and CEP290 mutations that lead to LCA. I mentioned AGTCs working on Stargardt disease and dry AMD. And Shannon Boy, through ITSINA and her lab at University of Florida, is also pursuing a gene therapy for USH1B. Just a little comment I'll make there. USH1B is a challenging condition to treat with gene therapy because it's a large gene. It's hard to put it in one of those containers I was talking about. But Shannon's developing a technology to split the gene and put it into 2 containers. And then when those containers arrive in the retina, they combine to form a full gene. So it's called dual vector technology. It's very cutting edge, and we're excited about the progress Shannon is making for that. That same technology could be used for Stargardt disease as well. Next slide, please. Okay, so we talked about gene therapy, gene replacement therapy, where you're moving or you're delivering an entire gene to the retina to replace that mutated gene. So gene editing, specifically through CRISPR-Cas9, is different. Instead of delivering a whole new gene, you're simply editing the gene that's mutated in the retina. Sometimes I like to refer to this as cut and paste. So you're delivering a pair of quote unquote molecular scissors to cut out the mutation. And in some cases, you may be delivering a tiny piece of gene to also help that gene function correctly, but you're not delivering an entire gene. So this approach is a little more targeted toward the specific mutation that's causing the retinal disease. Now, a collaboration between Editas and Allergan several months ago launched a phase 1/2 clinical trial for a specific mutation in the gene CEP290. Now, what's really important about this trial, it's the first time that CRISPR-Cas9 has been delivered to the human body to treat any disease in any part of the body. So there has been some CRISPR work done on cells outside the body, and then the cells are delivered to the body. But this is applying CRISPR-Cas9 directly into the retina of the affected patients. So they haven't reported results. They are also pursuing a clinical trial for USH2A for a region called exon 13. That's a hotspot in the USH2A gene where a lot of mutations occur. And I'll also note there's a lot of great CRISPR-Cas9 work going on in preclinical studies in other labs. I know the Foundation Fighting Blindness US, we have about 5 or 6 labs that we're funding that are pursuing CRISPR, including Hadassah in Israel, a lab in Italy, and there are others. So this is newer. It has the benefit of targeting specific mutations and helping people with these larger genes, which are harder to replace. So stay tuned as we get more of these approaches into clinical trials, and we'll see what happens. I think this is a promising approach. So next slide, please. So the next technology gets a little more complex in terms of how it works. And I'll try to simplify that discussion by saying that your cells, while they have DNA, and the DNA is the code or the genes that help your cells survive and do what they need to do, your cells have a messaging system to communicate that genetic information. And that messaging system works through what we call RNA, where that genetic information is communicated throughout the cell so that the cells know how to function and which proteins to make. So this company in the Netherlands called ProCure is targeting the RNA, the messages, not the DNA, but the genetic messages. And their technology, which is called antisense oligonucleotides, or AONs, I know that's a mouthful, but that technology is simply a small piece of genetic material to cover up the mutation in those genetic messages. And you can think of it as like a tiny piece of genetic tape to cover up the mutation in the messages. And the Foundation Fighting Blindness in the US is investing $7.5 million in this company specifically for their USH2A exon 13 clinical trial. But ProCure also has clinical trials underway for LCA, a specific mutation in CEP290. There were nice vision improvements reported in the phase 1/2 a few months ago, or I should say now actually several months ago. They moved into a phase 2/3. So we're anticipating additional results from there. ProCure, again, also has a clinical trial underway for USH2A mutations in exon 13. They reported some very early encouraging results, and they also have a clinical trial underway for the P23H mutation in rhodopsin, which is a common cause of RP. They're still yet to announce any results from that, so stay tuned. Also in development in the ProCure pipeline is the PE40 mutation in USH2A, some other LCA genes, Stargardt disease, a specific mutation there. They have a very active pipeline. These clinical trials are all relatively new, so they're doing a great job at accelerating their research and getting it into the clinic. So next slide, please. So we've talked about gene replacement. We've talked about gene editing. We've talked about targeting RNA with the genetic tape in ProCure. Optogenetics is yet another approach to gene therapy. So I'm really testing your capacity to understand gene therapy today, but this is all very exciting stuff. So on the right side of the diagram here, I have a side view of the retina. And at the top of this diagram are vertically oriented cells we call the photoreceptors, the rods and cones. These are the cells that make vision possible. They process light, they absorb light, they convert it into electrical signals which are sent to the back of the brain to create the images we see. So in people with retinal diseases, they lose photoreceptors. If somebody has an advanced retinal disease where they've lost all their vision, it's because they've lost all their photoreceptors. Now, the difficult news for somebody who's lost all their photoreceptors is all the genetic techniques I've talked about up to this point don't apply because there aren't any photoreceptors left to receive these genetic therapies. But here's the good news, the promising news, is that researchers have identified other cells in the retina, they're called ganglion cells, that survive even after someone has lost all their photoreceptors. In many cases, these ganglion cells survive. And I have an oval circling those ganglion cells. And what they're doing now in clinical trials is they're delivering a gene to these ganglion cells to make them light sensitive so that they work, quote unquote, something like photoreceptors. So it's a way of harnessing other cells in the retina for vision other than photoreceptors. What's nice about this approach is it's designed to work regardless of the gene that's causing somebody's vision loss. So this is gene agnostic. Now, there are 3 clinical trials underway: Allergan, GenSight, and another company in the US called Bionic Sight. And I won't get into all the details for sake of time on the different approaches here. They're all at a still relatively early stage, and we're still learning here. But there are other approaches to optogenetics which look, I think, at least technically speaking, even more promising, that will be moving into clinical trials. There's a company called Bayonne that has a molecule that they're moving toward a clinical trial. Another company called Videre, which we funded, that was recently acquired by Novartis. That technology is even more elegant than what's in clinical trials. So this space is really active. It's early, but it's really active. So I think there's good reason for hope, but it's early, so stay tuned. Okay, next slide, please. So now I want to move into the stem cell space. And I think it's important to understand that stem cells are still in an earlier stage of development. They're not quite as far along as gene therapies. But the real benefit of stem cells, there are 2 aspects of them that are very beneficial. One, you can make a stem cell into almost any cell type in the body. So you can grow new muscle, blood, heart, lung, and obviously in our world, we can make them into new retina, photoreceptors, and other cells in the retina. Now, the other benefit of stem cells is that you can make literally billions and billions of cells for a treatment from a tiny sample of stem cells. So they're easily replicated. You don't need many of them to make thousands and thousands of treatments. Now, there are a number of ways you can derive stem cells, and we've probably all heard of embryonic stem cells and other similar approaches. But what researchers are doing now, what's starting to move into the clinic, this is happening in Japan actually, they're taking mature blood from a healthy adult, skin or blood, and they're genetically tweaking those cells to revert them to a stem cell-like state. Then they're coaxing those cells forward to become retina, photoreceptors, RPE cells. We call this approach induced pluripotent stem cells. And in the future, you're going to see more and more stem cell therapies using this approach. We fund David Gamm at the University of Wisconsin for this approach. He has a company, a startup company called Opsis. They're working toward a clinical trial using this approach. So I'm going to talk about a few stem cell trials. And you can use stem cells basically in 2 different ways to treat the retina. One I call the protective approach or the preservation approach. More technically, we call it neuroprotection. And this is using a stem cell that is almost developed into a photoreceptor to try to save a recipient's existing retinal cells. So again, that's the preservation paradigm. And there's a company in the US called GenSight, which a few years ago launched a clinical trial of this approach using its retinal progenitors to try to save vision. So this is going to apply to people who still have vision remaining and are just trying to preserve what they have. So these cells are injected into the vitreous in the middle of the eye. They release proteins or growth factors that are designed or are able to keep the retina healthy. So they are actually now in a phase 3 clinical trial, or I'll correct myself. They're moving into a phase 3 clinical trial. They had excellent results in their phase 1/2, in their phase 2b. The treated eyes performed better than the untreated eye. So they're now moving into phase 3. And they've received some funding now from Santen, a big global pharmaceutical company, to help them move it through that later stage clinical trial. So let's move into the next slide. So another approach using a very similar cell type is being advanced by a company called Retinal out of the UK. And they have a trial underway at Mass Eye and Ear in Boston and a clinic in Phoenix. But instead of trying to preserve cells, the idea here is to replace lost photoreceptors, to restore vision. And instead of injecting these into the vitreous, these cells are injected subretinally underneath the retina. And the hope here is that these cells will actually connect with the patient's existing retina and restore vision. In their phase 2 trial thus far, many of the patients have had about 3 lines of vision restoration at 12 months, and now they're seeing that vision improvement has been durable out to 18 months. So they're expanding their phase 2 right now, trying to get more data with the hope that they can move into phase 3. The Foundation Fighting Blindness US provided some significant funding to get this trial off the ground, and we also fund that smiling gentleman on the right, Jason Commander. So for those of you with age-related macular degeneration, dry AMD or wet AMD, there's another stem cell approach that's targeting RPE cells, which provides support for photoreceptors. And in AMD, it's those RPE cells that degenerate first. So researchers are trying to replace those lost RPE cells. And there are actually a number of trials underway throughout the US and Europe to do just that. One of the trials is out of Southern California called, it's being launched by a company called Regenerative Patch Technologies. They are putting these RPE cells derived from stem cells on a scaffold, and they're transplanting it into the retina. That's what you can see sort of in this image of the retina on the right. And the hope is by putting in new RPE cells, you'll save photoreceptors. At University College London, they're using a similar approach. And again, there are other companies and labs targeting RPE cells for people with age-related macular degeneration. And I will note this approach may also apply to people with Stargardt disease. Next slide. So some other investments that are being made by the Foundation Fighting Blindness for some cross-cutting approaches include an anti-oxidative therapy targeting RP and other retinal conditions, potentially AMD as well. So this company is called Nacuity. In lab studies, we've learned that oxidative stress plays a key role in accelerating vision loss. So if you can mitigate oxidative stress, you have an opportunity to slow vision loss. Nacuity's oral molecule, their anti-oxidative therapy, is moved into a clinical trial in Australia for people with Usher syndrome. But hopefully, if the results from there are encouraging, they will launch a trial in the US for people with RP and Usher syndrome in 2021 as well. What's nice about this approach, it's designed to work independent of the mutated gene causing the disease. Now, another approach that we're providing funding for is being developed out of France, a company called Sparing Vision. And for those of you with RP, you know that your rods are affected first. Rods give you night vision and peripheral vision. And in advanced disease, after you lose rods, your cones start degenerating. And what the researchers in France learned is that there's a protein called rod-derived cone viability factor that's produced by rods that help cones stay healthy. That helps the cones stay healthy. So they've developed a therapy that produces this protein in an effort to preserve cones. So this will hopefully move into a clinical trial in 2021. And again, it's designed to work independent of the mutated gene causing someone's retinal disease. They'll be targeting RP and likely Usher syndrome, at least at some point, because as we know, Usher syndrome is RP with hearing loss. So again, hopefully we'll see a clinical trial in 2021 for this. That's the plan. Next slide. So I wanted to just briefly cover a lot of activity that's going on for Stargardt disease. And I'm not going to get into all the details of the 4 projects that I have mentioned here for the sake of time. But what I want people to understand is Stargardt disease is a condition caused by vitamin A processing. We all need vitamin A to see. We get it through our diets, and vitamin A is the magical molecule that makes our retinas light sensitive. You can think of it as like a fuel. But like a fuel, when it's processed, there are waste products that are produced. Now, in a normal healthy retina, those waste products are cleared properly. But in people with Stargardt disease, those waste products are not cleared. And when they accumulate in the retina, they lead to central vision loss. So what these companies are doing, Alkeus, Acusella, Iveric, Belite, and there are other companies moving into this space, they're designing therapies to reduce the accumulation of those waste products. So the companies I have listed here are at various phases of clinical trials. You're going to see more companies moving into this space. And this approach may also work for people with dry AMD. We're still at a relatively early stage for many of these companies, but stay tuned. And so finally, last slide. So I'd like to do a little shameless self-promotion, but just to give you a little background on me before I talk about my book here, is I've been in this space now for almost 16 years. And throughout my time in working with patients and families, I've always been inspired by the kids who deal with these conditions with such courage and enthusiasm. The kids are amazing. So while I often write about the research underway, I also wanted to take the opportunity to write a fictional story to honor these kids who take on retinal diseases so heroically. So I came up with this book, Retina Boy. It's published by Apprentice House out of Loyola, Maryland, in the US. And it's a story about a kid that's born blind. He's in Iowa. And I'm going to give you a little bit of a spoiler here. It turns out he's an alien from another planet of blind inhabitants who don't have retinas. And so you follow this boy and his human counterpart throughout the story. And eventually this kid, who's called Retina Boy, is called upon to save the Earth and this other planet. So it's a fun story. It's a story that's geared toward young adults, but I would encourage adults, grown-up adults, to enjoy it too. It's really fun for all ages. And I still have many free codes for Audible in the UK. I've used up all my US free codes. So if anybody is interested in a free Audible copy, send me an email, bshaberman@fightingblindness.org, and I will send you a free code. So I know we've gone a little bit over my time. Hopefully we still have a little bit of time for questions. Thank you, everyone.
Thank you, Ben. That was really, really good. And I would also endorse the book. I read it and I really, really enjoyed it. Anyone who is a music fan, they too will gain a lot from it. There's even a band in it called the Rhodopsins. So I would encourage people to check out that book. So we have a little time, maybe 1 or 2 minutes for questions. So Ben, one question that has come in is around the USH2A preclinical work that was mentioned, possibly the ProQR work. Is this work, this kind of approach, is it going to benefit both the vestibular and hearing element of Usher's, or is it just focusing on the visual aspects of Usher syndrome?
That's a great question. And right now, this work is all targeting the retina. So it's targeting vision.
Yeah. And then, yeah. And you also mentioned the RDCVF approach. How is that delivered? Is that an oral drug? Is that a gene therapy, like delivered like a gene therapy? How is that delivered?
It is a gene therapy.
It's a gene therapy.
It is a gene therapy, but it's not replacing anything or augmenting anything. It's simply a way to produce that protein in the cells. It kind of it empowers your cells to make that protein.
And we're all about empowerment, so if we can, exactly. And the work that you described on optogenetics, that truly is an exciting field, and we're so delighted and honoured that Professor John Flannery at the University of Berkeley sits on our medical and scientific advisory board, and I know he's a great contributor towards Foundation Fighting Blindness US as well. So there's huge talent out there and researchers dedicated to the cause. One of the things that we've noticed is a lot of the work described seems to be very much, with the exception of optogenetics, gene specific. And we know that, you know, maybe 40% of cases or individuals will not receive a genetic diagnosis because the genetic cause has not yet been identified. What kind of work is ongoing to try and find these new genes that we don't yet know?
That's a great question. So one thing to keep in mind is that, yes, we need to find new genes, but genes can be mutated in ways that are hard for genetic testing to find. So your gene may be common, but it may be mutated in a way that's difficult for genetic testing to pick up. So we're actually funding a project called the Elusive Genes and Mutations Project, which includes Steve Dager, Rada Ayagari, and Mass Ioneer. We're investing about $2.5 million in that project to find those elusive genes and mutations. And that work also is occurring in other labs around the world. You look at places like Nijmegen in the Netherlands, Institut de la Vision in France. And every year we get better and better at identifying the mutated genes. Right now, in most cases, or I should say collectively, we can identify the mutated gene in about 2/3 of the cases. But one last little comment, many of the therapies I talked about today, the stem cells, nacuity, sparing vision, those are all designed to work independent of the mutated gene.
Exactly, and as you say, there's a lot of work going on. We ourselves are funding very similar to what your Elusive project is, and it's a discovery arm of Target 5000, and that's a collaboration between Trinity College Dublin and the group in Nijmegen in the Netherlands. And it's really important that we continue searching for those unknown causes of retinal degenerations.
Definitely.
Just one final question, and you know, it's geared around your book. Can I just ask if the audio version is available via audible.com app? Do you know? Or will we follow up separately?
It is an Audible book. That's the only way it's available. So again, if you want to buy it, I'm not going to stop anybody from purchasing it on Audible in the UK. But I'm also happy to give you a free promo code so you can get a free version of it as well.
Brilliant. Look, thank you so much, Ben, for joining us today. And again, unfortunately, we don't have as much time as we would love to give to this aspect, but be reassured that we are coming back to you very soon, early next year, with a whole suite of condition-specific webinars where we will look at the research and clinical trials and support for each of those. So thank you very much, Ben. Hopefully we can bring you back to Dublin. And
I'd love to come back.
Yeah. And Hoth is beautiful. Hoth is definitely beautiful.
It really is. All of Dublin is beautiful.
It's very quiet at the moment, so we'll wait until things improve.
Okay.
So thank you, Ben.
Everybody. Okay.
Thank you.
Bye-bye.